Welcome to Tulliome's September Nature and You lecture. My name is Jeff Ben. I'm the Education Associate at Tulliome. If you don't know, Tulliome is a conservation nonprofit based in Woodland. Our mission is protecting public access to public lands and educating folks about the natural history of those public lands. Our area of focus is the northern inner coast range, which are the mountains that are off to the west of Davis and Woodland, with a particular emphasis on the Berryessa Snow Mountain National Monument, which is the relatively new national monument that stretches from kind of the southeast corner of Lake Berryessa up through the Cache Creek Wilderness and into the Snow Mountain Wilderness up in the Mendocino National Forest. We do lots of different events. So we do K-12 field trips, we do hikes and trail building days for the public, as well as this monthly lecture series. We're also really involved in advocating for expanding the Berryessa Snow Mountain National Monument by adding the Moloch-Loyok region to it. That's a ecologically and culturally important ridgeline that runs along the border of Lake and Colusa counties. So if you're interested in learning more about any of our activities or advocacy work, you can do that at our website, tulliome.org. So without further ado, I will introduce our speaker for tonight. So Dr. Laura Bogar is coming to us from UC Davis. She has been researching the relationship between mycorrhizal fungi and plants for quite a while now, going all the way back to her undergraduate days at Lewis and Clark College, where she got her bachelor's degree in biology. She then went on to Stanford University to earn a PhD in biology, and then did postdoctoral work at UC Santa Barbara. She now has her own lab at UC Davis in the Department of Plant Biology, where she studies the relationships between plants and fungi and how that all affects the ecosystem. And so we'll hold any questions that you might have until the end of the talk, and then you can either send them in to me through the chat, or you can just unmute yourself. And so with that, I will turn it over to Professor Bogar.
Thank you so much, Jeff. This is going to be great. So my goal is to spend about the next 45 minutes talking with you about a couple of different facets of plant fungal symbiosis, and then we can take some time for questions before the top of the hour. Is that about right? Let me know if I've got the timing wrong. Okay. So as has been said, I work on symbiosis between plants and fungi, especially tree roots and soil fungi. I see a couple of my students in the audience, so this will be very familiar to them. And I've entitled today's lecture, Symbiosis in the Soil, Trees, Fungi, and Their Underground Networks. And this slide, we're going to be seeing a lot of in the background. Can you guys see when I move my mouse on top of the slide? Great. So here we have a mushroom. In my cartoon, I imagine this as a mushroom in the genus Suillus, which you might see around Northern California under pine trees. And this Suillus mushroom is attached through its hyphae to a mature pine tree. And we'll be thinking about how they interact today. So my main goals are fairly high level. First, I want you to walk away knowing at least a little bit about what I think is cool about roots and fungi and their interactions. So my goal is to hook you on some of this. I also want to give you a sense for how scientists study this kind of thing. I'm really interested in a lot of facets of plant fungal interaction, and especially resource movement between trees and fungi. So I'll give you a window into how we look at those questions in the lab. And finally, I alluded to fungal networks in my title because they're a big object of public fascination right now, and that is because they're really cool. I want to take a minute with the group and walk through what we know about how forests are connected below ground, where the missing links are, and like where the exciting research needs to go. Okay, what's cool about roots and fungi besides everything? Let's start with the fact that it's almost ubiquitous. Nearly every plant needs fungi on its roots to survive. This photo is an oak woodland outside of San Jose. I went backpacking there a few years ago, but it's a familiar landscape to those of us in the Davis area. We have oaks, pines, grasses. If you look out at the landscape, the obvious things that all of these organisms are doing might be photosynthesis and swaying in the wind, but all of these plants have roots in the soil, and those roots are associated with fungi, many different kinds, and those fungi are mediating the plant interactions with the soil and the resources there. And I'm going to be using this word symbiosis a lot tonight. This may be a concept that is familiar to you. It may be a new idea, but I'm really just talking about living biosis together, sim. And we're thinking especially about fungi and plants living together. This is an old, somewhat famous at this point, image of a pine seedling engaging in symbiosis with a mycorrhizal fungus, which we'll get into in a moment. And you can see that the symbiosis is really extending the reach of the plant's root system and allowing it to access parts of the soil that it couldn't access by itself. So what do the plants and the fungi gain from this symbiosis? Well, plants, we know, are really great at taking carbon out of the atmosphere and turning it into high energy carbon molecules, things like sugars. And in the symbiosis, the plant sends some of those sugars down to the fungi on its roots, where the fungi can use it to build things like mushrooms. In exchange, the fungi grow into the soil and gather resources like nitrogen or phosphorus or even water and bring it back to the plant so the plant can use it for its own purposes. This movement of resources is obligate for these organisms. And by that, I mean, you are never going to go into the woods and find an ectomycorrhizal mushroom without a host plant. It just doesn't happen, although we can grow them in the lab on petri dishes without a host. And similarly, in the forest, you are not going to find a successful adult pine tree without ectomycorrhizal fungi on its roots. Although in the growth chamber, we can grow these seedlings without fungi, and if we baby them, they can survive for a little while. And this resource exchange occurs at this specialized structure called an ectomycorrhiza, which we're going to explore a little bit. It's a lot of syllables, and I want everybody to be comfortable with it as we go through. It's three parts, this word. We'll start with the most important ones, which are myco and rhiza. I've kept ecto in parentheses, so don't think about that right now. So myco might be familiar from mycology. Myco is fungus. Rhiza might be familiar to those of you who've studied some botany. This one means root. So we have a fungus root. And this mycorrhiza word is the one that appears all over the plant tree of life. Nearly every plant engages in some kind of mycorrhizal association. Ecto brings us into my favorite little corner of plant fungal symbiosis. This is a specialized kind of fungus root relationship where the fungus stays on the outside of the root, the ecto part. So this is an image, a photo I took of an ectomycorrhiza to give you a sense for what we're talking about. This is my thumb. This is a pine root, a bishop pine from Point Reyes. And you can see the terminal fine roots here have become swollen. They're covered in white stuff. That's all hyphae. This is a symbiotic structure. If you were to zoom in on this, you would see in greater detail the way that the roots often branch in unusual ways once they're associated with the fungus. This helps optimize the surface area across which the resources can move. The hyphae themselves extend into the soil to maximize resource uptake. And if you were to take a cross-section of one of these root bits, you would see the hyphae penetrating in between the outer cortical cells of the root. In the ectomycorrhiza, these hyphae always stay outside the cells. They're like the world's tightest toe sock on the plant. And this is how the resources move between plants and fungi. And it can actually be kind of a lot of resources. It's been estimated that plants send often up to 20% of their photosynthate to the organisms on their roots. This is a pine seedling associated with fungi that have been given some nutrient patches over here. And the pine seedling was labeled with radioactive carbon dioxide, which we're going to visualize as a heat map so you can see where the plant put the carbon that it fixed through photosynthesis. And I love this image because it's striking. The plant, of course, makes sugars and uses a lot of those sugars end up in the leaves. But you can also see incredible hotspots where the fungi were growing, respiring, taking up resources from the nutrient patches. So the plant and the fungus are interconnected profoundly when it comes to especially carbon. And these mycorrhizal symbioses are really ancient. Here to our cartoon, I have added a little grass, and it's associated not with the mushroom, but with these little orange fungi with some globs on the end. These are my artistic interpretation of an arbuscular mycorrhizal fungus. This is the ancestral type of mycorrhiza that it's thought the original land plants associated with these kinds of fungi to help them liberate nutrients from the soil. So this emerged about 400 million years ago. The ectomycorrhizal symbiosis between these often bigger temperate trees and large-bodied fungi arose about 100 million years ago. So it's a much newer thing. And the ectomycorrhizal symbiosis has actually evolved independently many times from decomposer lineages. So a fungus through evolutionary time will start out growing in the soil by itself, eating mostly dead materials, and eventually these lineages figure out that plant roots are a powerful source of sugar, and they learn how to do symbiosis with the plant roots. So I've put some images of mushrooms on the slide here. These are very closely related. Maybe they're familiar to you. I would love to see in the chat which one of these you think gets its energy from decomposing, because the other two are ectomycorrhizal. If I'm able to see the chat. Oh, look. Oh, we have some votes coming in. Okay. Okay, there's some cheating happening with some identification, but I respect it. It's an engaged audience. Okay, great. Thank you very much for participating in our informal interaction here. Yes, so indeed, option one is the Saproamanita thiersii, as Christine identified. So that one is a relative of the mycorrhizal Amanita taxa that we see in 2 and 3, but this one, number 1, can grow all by itself without a host plant. So it's remarkable. Fungi have done this at least 76 times independently through evolution, and they're probably continuing to do it under our noses. Another important aspect of the ecology here is that plants can have many fungal partners at once, especially in ectomycorrhizal symbiosis. They might have dozens of individual fungal species, potentially hundreds of separate individual fungi on their roots simultaneously. Here I have added a cartoon Amanita to our pine root system to compete with the Suellus that was already here. And the fungi that are on a plant's root system can be super diverse, both taxonomically and morphologically. These images are mushrooms that I have found in collecting expeditions in California over the years. You see profound variation in morphology, like the structure, the color, edibility. A couple of these are choice edibles, some of them are definitely not. So chemical variety, and it remains unknown the extent to which this obvious diversity might map to diversity in symbiotic function. So to what extent does it matter to the plant that it has some of these really tasty edibles and other brightly colored, very bitter things? Like what are the symbiotic differences there? And that's one major research area that we try to peck away at basically with every project we do in the lab, is trying to quantify the traits of these fungi in symbiosis and how they differ from each other. The final wrinkle I want you to keep in mind here is that these organisms can be specialized or generalized, as is true in many symbioses. So the fungi generally will be specialized to some extent on certain plant hosts. For instance, ectomycorrhizas themselves, it's not all plants. It's generally some big woody trees and some fungi evolve from decomposers. So that's a specialization in itself. And the fungi often will associate, for instance, just with pines or just with Doug firs. But there are generalist fungi. This little brown one that I illustrated is representing a common fungus I work with that you'll see in a few slides, the Lephra terrestris, and this one can grow with pines or oaks, whatever. It's pretty broad. The plants, on the other hand, are usually generalists. They can often associate with most of the fungi that they encounter as long as they are ectomycorrhizal. So we've got a little bit of asymmetry in terms of specialization. And these interactions between big woody trees, cool, diverse mushrooms are not only fun to think about because we like being in the woods and looking at mushrooms, they also have important implications for how resources move across landscapes. So you all may be familiar with the fact that carbon dioxide can go into trees, and that's an important part of how we can try to manage the carbon cycle as we keep pumping carbon dioxide into the atmosphere. The trees, as you've just seen, put some of that carbon into the fungi, and both of these organisms have to respire and put a portion of that CO2 back into the atmosphere. So thinking about how fast this carbon cycle goes, the trees and fungi are a key part of the equation. Similarly, nitrogen cycles through forested landscapes. It comes in through rain and runoff from agricultural fields. It can be taken up directly by the trees or by the fungi. It can be traded from the fungus to the tree, and it also can cycle back into the atmosphere. So to really understand how these resources are moving, I think it's important that we understand the processes controlling resource movement between the trees and the fungi. They're really at the nexus of many different cycling resources. So we can really bring it from a single root tip, I hope, to understanding what's happening in the atmosphere. So we're going to talk about sort of two major questions today. First, I'm going to show you some data because one of my goals here is to illustrate for you how we try to untangle some of these questions in the lab and really get you oriented to the processes that we use to learn about them. The university is also very committed to energy conservation, so my lights will go out every once in a while, but I'll just wave them back. So you'll see some data. We'll think about how resources move between plants and fungi. In the second part of the talk, we're going to think about how fungi connect forests below ground. I'm going to walk you through some evidence that's been compiled by other colleagues to really think about what we know and what we don't know, and show you where the interesting gaps are and where I hope you'll be inspired to look more and maybe suggest some experiments we could do. So we'll start with part one. How do resources move here between plants and fungi, and how do we study it as scientists? And the more specific question is whether plants can reward helpful fungi by giving them carbon in exchange for nitrogen. So I'm interested in this symbiotic cooperation, and I want to know whether plants can reinforce it by demanding nitrogen from the fungi before giving out that carbon reward. What would rewards look like? What do I mean by this? So if Sue Willis is bringing a large amount of nitrogen to the plant, and Amanita, for some reason, is bringing only a little bit, then my definition of rewards is very simple. The plant gives carbon in a large quantity to the fungus that brings back a large amount of nitrogen, and only a little bit of carbon to the fungus that's bringing a little nitrogen. So just reciprocal resource movement. So to answer this question, I conducted some experiments in graduate school using split root pine seedlings. And I would put different things on the sides of the root system to give the plant a choice between treatments. So this pine had nitrogen on one side of the roots and just a sandbag on the other side. So you see these little nylon mesh bags. I put resource patches in there. And the result is that one side of this root system has a lot of tasty stuff, and the other side of the root system is really kind of worthless from the plant's perspective. And the plant has fungi on its roots here. So this fungus can bring nitrogen to the plant, and the plant, ideally, if it does rewards, would bring carbon back to that fungus. And on the other side, it would not be such a strong trade. To conduct this experiment, it wasn't just a sand versus nitrogen choice. I also had microcosms with sand on both sides of the root system and some with nitrogen on both sides of the root system so that I could tell how variable it is baseline, even when the two sides are the same. And I also had plants with no fungi on their roots. You may be aware, if you grow plants yourself, that plants are very good at taking up nitrogen directly from whatever you grow them in. This is what plants do. So they don't always need fungal help, and it's important to keep track of where they allocate their carbon, even if there's no symbiont involved. And to carefully track where the plant was putting its carbon resources, I used a technique called stable isotope enrichment. And you may recall, if you've taken chemistry, or maybe this is new, there's a couple of different flavors of many different atoms in the universe. You can have these different isotopes. And as long as the number of protons is correct, then you have basically the right chemistry. But if you play with the number of neutrons, you can end up with heavier or lighter atoms. So the most common form, the most common flavor of carbon is carbon-12 here. The least common one, or not the least common, but a much less common isotope is carbon-13. It does occur in nature. It has a natural abundance that is fairly low. Importantly, it's not radioactive. So when you think about isotopes, you often think about radioactivity, and that's not what this is. It's a stable isotope that is naturally present. So this might be a cartoon air sample of just your living room air right now. And I wanted to be able to keep track of where plant carbon was going. So I had the plants photosynthesize in an atmosphere with way more carbon-13 than you would ever naturally have. So this allowed me to know the sugars the plant made while it was in that atmosphere were going to be heavier than the sugars it made at any other time. And I can track that pulse of heavy sugar down into the roots and see where the plant was putting its carbon at that moment. So these are the little plants in their labeling chamber. This is a slightly different experiment. You'll see there's an oak in there, but it's the same chamber setup. And they sat in there for 8 hours photosynthesizing in their carbon-13 enriched atmosphere. And I looked to see whether the plants put more carbon towards the side of their root system that had a nitrogen patch, and whether it made a difference if there were fungi there. So what would I expect to see if the plant rewarded nitrogen resources by providing carbon? On the y-axis, we have plant carbon. On the x-axis, we have nitrogen coming into the plant. And if there's no rewards, if the plant just dumps carbon into its root system indiscriminately and it doesn't matter how much nitrogen is happening, I would expect a flat line here. If there are rewards, I expect a positive relationship where the more nitrogen a root is bringing in, the more carbon it should have. And with the fungi, we might have a slightly different relationship than without the fungi, maybe more extreme rewards for fungi. What did the data show us? These are basically the same X and Y axes as we just had. We're looking specifically at the difference in carbon between these paired root compartments so you can tell like relatively how much more carbon is one side getting than the other. So the y-axis is C, the x-axis is the difference in nitrogen. So do we have a positive relationship? I also distinguished between fine roots without fungi and ectomycorrhizas with fungi to see if the plant was treating its symbionts differently than its own tissues. And it wasn't at all, but we do have strong signal of rewards. So the plant does invest a lot more carbon into roots that bring back nitrogen. But at least in this experiment, it did not matter at all if those roots were attached to a fungus or not. It was exactly the same process, which is kind of encouraging in that the plant doesn't have to do anything fancy necessarily to accidentally reward helpful fungi. If it's already rewarding its own roots for getting nitrogen, this could be an easy process. So we learned that rewards are possible, but it was a little confusing that the roots and the fungi looked exactly the same. And I wanted to know how fungal competition might change things. So I added in another fungus, and I also changed up the nutritional setting a little bit to get at some subtleties in these processes. So I did another split root experiment. In the end, trying to figure out whether there might be a resource marketplace. If we do have carbon for nitrogen trading, is there usefulness in thinking about this more broadly as a plant having carbon, giving it in direct exchange for resources? So for this experiment, I used two fungi that you've seen cartooned, but not in real life yet. This is the Lephra terrestris. It's a generalist. This is Suillus pungens. It can only grow with pines. They're both common in California pine forests, and they often grow on seedlings. But as I mentioned, they have different ecologies. So for this experiment, it was still split root plants. The Lephra and Suillus could compete indirectly on different sides of the split roots. Or you could have the same fungus on both sides of the root system. or no fungi on one or both sides. And we also had 2 different fertilizer treatments. We had high nitrogen or low nitrogen to see whether the fungi and the plants behaved differently in sort of a richer atmosphere. So we're looking at competition both between fungi indirectly, the fungi are not touching, but they're indirectly competing on the roots, and between roots and fungi directly for whatever nitrogen we're putting into the system. This is what these new split root pots looked like. It was a step up from the first experiment. You can see we have our fungus-only compartment bolted onto the outside here. The plants are poking out of the tubes and their roots are split between these 2 sides. And if you were to open up those split root compartments like a book, you would be able to see how we use these for 13C and 15N labeling. So we used the same principle that we did in the last experiment, but we also use a stable isotope of nitrogen to pump in a label that I can track into the plant. So we have a fungus-only compartment and a root-only compartment, and they're, the 2 halves of the root system are separated by a solid barrier. There's a mesh barrier between every root compartment and fungus compartment so that you can have the fungi grow through, but the roots are excluded. And this allows me to add nitrogen that only the fungus can access when I need to. So the 15N goes right into the fungus. And the 13C, of course, the fungus can't photosynthesize, so 13CO2 is safely taken up by the plant and given to the fungus. So Thelephora versus Suillus, which fungus was a better partner? We're going to look just at total plant biomass at the end of the experiment. These seedlings grew for about a year as a proxy for overall seedling fitness. It is not a perfect facsimile for what would happen in nature, but if a plant is bigger, it's done better in our experiment. And we're going to look at these data separately for the high nitrogen fertilizer and the low nitrogen fertilizer. On the x-axis, we have fungal treatment, which just tells you which fungi were on the split root. You could have no fungi versus no fungi. You could have Suillus versus no fungi, Suillus versus itself. Thelephora versus Suillus is that key competition treatment. Thelephora versus no fungi, or Thelephora versus itself. Every dot is a plant. And you can see that Thelephora, with more Thelephora, made for some very big, happy plants. That was a very effective mutualist in this experiment. You'll also notice we had reduced replication anytime Suillus was there. Thelephora is well known as a greenhouse contaminant, and it did indeed contaminate many of these treatments in this experiment. So we know a lot more about Thelephora now than we do about Suillus, but I think it was still informative. In low nitrogen, the pattern was not as clear. The plants all grew to about the same extent, and they were pretty nitrogen stressed. So we have some really interesting contrasts showing up already in these 2 different nitrogen situations. Thelephora being very helpful in high N, not helpful in low N. So what did that mean in terms of resource movement? Was the plant able to prefer Thelephora in this high nitrogen situation where it was helpful? So here we're going to look specifically at how much carbon the plant gave to Thelephora. We know Thelephora was very helpful in the high nitrogen treatment. So we're going to look at the carbon allocation ratio to Thelephora. That is, in a split root system where you have Thelephora on at least one side, did Thelephora get more carbon than its competitor or less carbon than its competitor? And it could be competing versus Suillus or no fungi, or it can compete versus itself. And you can see that in high nitrogen, where we saw Thelephora being a very useful partner, Thelephora got a little bit more carbon, and it did get more carbon than its competitor generally, but it's not quite statistically significant. It's not a super strong effect. When Thelephora competed versus itself, both sides of the root system got almost exactly the same amount of carbon. So that's encouraging. The plant wasn't distinguishing between those identical treatments. In low nitrogen, intriguingly, the plant preferred Suillus. We didn't see the Suillus plants get bigger in low nitrogen. It's not clear why Suillus got more carbon, but it sets up an interesting mystery that I'm hoping to follow up on here. So it does seem like plants might have some ability to sort of comparison shop if this is a marketplace, but it's not super consistent or super strong. And some of these resource allocation patterns are not consistent with plant benefit. So the other question is about fungal competition, thinking about using a marketplace analogy to motivate some hypothesis forming here. Did the plants get a better deal when fungi competed, just like you might get a better deal on your groceries if there's a few grocery stores in town. Here we're going to look at the exchange rate, carbon for nitrogen exchange rate in Thelephora mycorrhizas, because again, we have a lot of data on Thelephora from this project. The competition treatments are as before. If you have a high value on the y-axis, it means that you were getting expensive nitrogen from Thelephora, you being the seedling. And if you have a low value, then the seedling was getting cheap nitrogen from Thelephora. So we'll see how that rate changed. In high nitrogen, when Thelephora had some kind of competitor, it seems to have offered a somewhat better exchange rate to the plant, or the plant was able to demand a better exchange rate. You had a better, cheaper nitrogen. And in low nitrogen, you don't really have the same effect. It's pretty much the same pricing across the board. And that might be because nitrogen was just so limiting in that treatment. We hit the floor of what the plants and the fungi could survive. So yeah, under high nitrogen, having a competitor elsewhere on the root system did change the trading dynamics and give the plant a better deal for the nitrogen. Under low end, we didn't see it. So under some circumstances, maybe maintaining diverse fungi could help a plant get a better deal on fungal nitrogen by sort of bargaining them against each other. But the fact that the pattern is inconsistent means that this analogy is not capturing all the dynamics that we need to. So, okay, that's the resource trading data story. What have we learned? Is there a resource marketplace with rewards for cooperation? We learned that plants, or at least these pine seedlings, sometimes reward helpful fungi, but not always. And we don't have resolution on the factors driving the unexplained resource allocation patterns. Whatever's happening here, it's more complex than carbon for nitrogen. I mentioned in the introduction that resources like phosphorus, water, potassium, magnesium, there's all kinds of resources that might be trading here. And it's also true that I think processes like sourcing dynamics are likely driving a lot of these processes in major ways that can't be perfectly explained by trading metaphors. So this is the kind of thing that I'm planning to follow up on the next couple of years. We're going to build a stable isotope enrichment chamber here at Davis. It should be very productive. But in our last 15 minutes or so, I would like to sort of change gears. I've got us in a new slide theme because we're thinking about a new set of ideas. And we're going to move away from data that I have produced and into data that are in the zeitgeist. I want to talk about fungal networks. How connected are forests below ground? Have you heard of the Wood Wide Web? If you're interested in fungi, you may not be able to navigate the culture without thinking about this a little bit. And if we were in person, I would have you shout out your definitions of the Wood Wide Web. But seeing as your Zoom faces right now, I will not make you do that. This image is an example of a hyphal network. I guess it's not connecting multiple plants, but we have pine roots, the brown stuff, and the white is all Suillus hyphae growing from the pine roots out into the soil. And you can imagine those connecting multiple plants. So a lot of people will say something about a network of fungi connecting trees. That might be part of it. There's often sort of a sense of the forest as internet, maybe information passing between the trees through the fungi, that kind of thing. The possibility that maybe the Wood Wide Web helps seedlings survive. So today we're going to be thinking a lot about these questions in the context of a paper that came out recently from Justine Karst, Jason Huxma, and Melanie Jones. And there's also been several other papers since then examining some of these ideas and revealing both where they're supported and where there's gaps. And so I do recommend checking out those papers if you get a chance afterwards and you're interested. So we're going to think through what the Wood Wide Web requires if it's able to do all of these things. My thought is that my thought and based on the scholarship of these excellent other scientists. So what we need is first, trees have to have fungi on their roots for any of this to happen. That one I feel pretty safe about. We need one tree to be able to associate with many fungi. I've already shown you that also seems pretty safe. We'll get into that a little bit more. Not only that, though, we also need each of those fungi to potentially be able to associate with many trees. So we'll think about that. And that's going to be really critical if any sort of information is going to pass through a fungus from tree to tree. We also would need information to move from one tree to another if it is in fact an internet of trees. And ideally, if it's a fungus network doing it, that information should move through fungi. So thinking about all of these requirements together, question one, do trees have fungi in their roots? Absolutely they do. I encourage you to dig around underneath a pine tree the next time you get a chance, because their roots are fairly shallow. You'll probably find some mycorrhizas. So that's great. Can one tree associate with many fungi? I asserted to you that this is possible, but I want to show you a nice figure just to sort of show you this is, it's not a tree, it's an Arctic herb, but I think Ella Thoen did such a nice job with this paper, looking at Bistorta vivipara, like these weird little herbs that have ectomycorrhizal fungi, but it's basically a tiny tree for our purposes, in the sense that it has the same kinds of fungal associations. She literally mapped the identity of like basically every root tip on these little plants and found so many fungal species. Each color, you can't really read it. Each color is a different fungal species. And you can see they're hardly even clustered. They're all over the place. So if even a tiny Arctic herb can have dozens and dozens of different fungi on its roots at the same time, yes, entire trees can have dozens, hundreds of fungal individuals. So that part is not a problem. Thinking about whether a fungus can associate with several trees becomes a little bit more complicated. Partly because fungal individualism is hard to define. So here it's our familiar sort of tree fungus cartoon. I've added an oak seedling to have a network in place. And we have a generalist fungus connecting our oak seedling to our mature pine tree. So because we can see underground with X-ray vision in this figure, it's very clear that the fungus is connecting the oak seedling to the pine tree, and there's the potential for resource movement or maybe signal transfer between them. But thinking like an ecologist, what else is happening in the soil? We have all kinds of weird little bugs. We've got disturbance events. We've got mycologists out there with their soil sampling tools, digging trenches. So if you have a bug that happens to crawl through and eat a chunk of your mycelium, it's going to look exactly as if these two plants are connected. If you happen to sample over here and sample over here and sequence the fungus, genetically it's an identical fungus, but it's no longer physically connected because a bug crawled through and ate it. And convincing yourself that there has been no severing of the hyphae is really, really hard. So you could have an identical genetic individual that may not be physically connected because of recent disturbance, and they can survive as identical genetic individuals for a very long time. When people do map these genetic individuals, it's very cool. This is a figure from a paper that's quite old now, but I think it's really nice. They did go out and identify genetic individuals, which is much harder than identifying just what species of fungus is, which is what I usually do. So they identified genetic individuals of the fungi, and the dots show you mycorrhizas that they sequenced, so the little roots with fungus that they took out of the soil. And the genetic individuals are connected in the shaded zones. So you can see there's this bright green individual that's like way all over the place. The blue guy, it looks like an octopus. So these fungi genetically are identical across really big and weirdly shaped areas. The lines are tree roots. You can see how the genetic individuals connect back to their source tree because they identified also which tree individual these things came from. It's a beautiful map, and it really shows us the ways in which this patch of forest is connected. But it doesn't answer the question of whether these genets are continuous. We don't know how many weird little soil bugs have crawled through to disrupt these hyphal connections. And that's even harder to do. So these maps are really hard to make. We also don't have very many maps of genetic individual fungi in individual forest patches. So because identifying the tree individual is hard and identifying the fungus individual is hard. So to my knowledge, there have been 5 done in 2 tree host species. And you can find the same fungal genotype separated by up to 21 meters. But finer scale studies on this kind of thing have found that they're only continuous for up to 40 centimeters. So that's a much smaller area than what we saw with these big giant patches. These can't be continuous if indeed continuity tends to break down at 40 centimeters. So I think there's room to do a lot more work on this. I would love to know the extent to which individual forest patches are connected by continuous networks or discontinuous networks, and at what rate these form and break. So I'm going to put this with a check question mark. One fungus can associate with several trees, at least transiently, because we see these genetic individuals moving from tree to tree, but we don't know how long that remains a continuous connection. The fourth requirement would be that information could move from tree to tree. So this is kind of a separate question from the fungal continuity thing. We know that trees can respond to information from other trees. For instance, if a tree is shading another tree, the shaded tree is going to respond strongly to that. Root competition is another thing that trees are known to respond very strongly to. They can sense the presence of roots, excrete chemicals. Litter chemistry can make a big difference. There's also been some really cool studies looking at volatile response in trees. It's been called the talking trees, which I think is somewhat contentious, but it certainly is true that if trees detect the smelly volatiles coming off of another tree that's getting chomped by herbivores, there are some trees that can respond by upregulating their defenses against those herbivores. So there's a lot of information that can move between trees. But does that information move through fungi, or is it something else? So this is another photo I took of a pine root with some active mycorrhizal hyphae. This is the Lephra terrestris, which you've been seeing as a cartoon. So we know that it's theoretically possible that some information could move through hyphae if indeed they're connecting trees for some period of time. But to my knowledge, this hasn't actually been shown. There was a study years ago now that showed that ectomycorrhizal fungi could move water and dye. It wasn't terribly far, but it was from a big tree into a little seedling. But the extent to which that happens is uncertain. The dye did not move into every seedling in the area. It was like one or two root tips from one seedling. And I think there's a lot to explore about what kinds of molecules might move. It's a little bit tricky. That symbiotic interface I showed you where the hyphae grow into the plant root, that's a very refined organ for controlling resource movement. So thinking about which kinds of molecules would move across that interface into a plant is interesting. Things like simple nitrogen forms, important for symbiosis, yeah, those totally move into the plant. What about other stuff? Unclear. So I think there's a lot of interesting research to be done, especially in actual field context, because we can do any number of things with common mycorrhizal networks in growth chamber experiments where the networks are not being disrupted by natural forces. But I think it'll be interesting to try to do some of this in field contexts and see what happens. So this one gets an X question mark from me. I think there's still a lot we need to look into. In the end, does the wood wide web exist? I think trees absolutely could be connected by shared fungi, maybe frequently, but we don't know how long those connections might last, and it's really hard to demonstrate in real time when it's happening in the field. We also know that plants are sensitive and respond to information in the environment. But it hasn't yet been shown that fungi are the medium communicating information about nearby trees, because the soil medium is so complex. There could be water flowing through the soil to move information around. There could be volatiles percolating through air pores, and also fungi. There's many, many different ways that plants are connected. And thinking about the mother tree hypothesis, it's true that sometimes seedling performance improves from being planted under a tree that has a good mycorrhizal network. But it's not consistent, sort of similar to the evidence for plant rewards that I showed you. We have really context-dependent outcomes. Sometimes seedlings are helped by these established mycorrhizal networks. Other times they're not at all, or maybe it makes things worse. So I think Justine did a nice job sort of walking through the relevant field studies on this, and I would recommend checking it out. I think it's a really complicated situation, and I think there's a lot to learn. So we don't have to throw out all of these hypotheses. I think they're really interesting. But what do we have if we don't have the Wood Wide Web? I do believe that trees and forests are connected in fascinating ways. There's water movement through the soil, volatile chemicals in the air and in soil pores. We have pollinators and herbivores visiting these plants. Some species engage in root grafting, where their roots literally grow into neighboring plants, and that is a very clear physiological connection. And I think fungal hyphae probably sometimes do connect neighboring trees, but quantifying the magnitude and the impact of those connections is a really critical open area of research. And these connections can facilitate not only cooperative mother tree type interactions, but also competition directly between plants. So if you'd like to know more, I do recommend there's a link I can send out of Justine's talk. But I'd love to talk through whatever questions you have about this sort of thing at the end of the session. So if you recall, our goals today were to engage you with the things that are cool about roots and fungi. I hope that you have at least a few of those to take home. I showed you how scientists study resource movement between trees and fungi, or at least how one scientist has done it, showed you some data, got you some graphs, and we thought about how connected trees and forests are underground and where the knowledge is and what the key gaps are. So with that, I'd like to thank you very much for your attention. Thanks to my funding sources and the students who were involved in this work. Thanks very much to the students who are involved in our ongoing work, who are on the call right now. And I'd be delighted to take any questions.
All right, do we have any final questions for Dr. Bogar? All right, so we'll wrap it up here. Thank you so much for a great talk. If you are interested, this is a monthly seminar series. We've got our next talk coming up in October. That's going to be with Andrew Folks, who's going to be talking about hikes and trails in the Berryessa Snow Mountain National Monument. So if you want some good tips as you're heading into fall hiking season, be sure to tune in for that one.